very much. So today I'm not going to be telling you about my research. I'm going to be helping you to hear about other exciting research that that is going on in this field for people with PSP and CBS or CBD. So the first talk that we have is Dr. Maura Malpetti. So she's a Race Against Dementia and AR UK Fellow at the Cambridge Centre for Frontotemporal Dementia, which includes PSP and CBS. And she's interested particularly in the neuropathology and biomarkers of these diseases. Today, she's going to be telling us all about brain scans and blood tests to track inflammation, and there'll be time for questions afterwards. So she'll talk for around 20 minutes, and then there'll be time for questions afterwards for around 10 minutes. So please put your questions in the Q&A box. Okay, Maura.
Thank you so much. Can you see my slides on the screen? Okay, excellent. Thank you so much, Boyd. And thank you so much, Rebecca, Megan, and also Nigel to host this fantastic meeting. It's so good to be here and show a bit what we are doing in the lab in the context of our research in PSP and CBS. Yeah, as Boyd mentioned, I'm the first researcher here presenting today, and I'm working at the University of Cambridge, and the PSP Association has been very crucial in the research that I'm doing and trying to expand as well. So I will show you a bit of our research on brain scans and blood tests to track inflammation. And there are already many words that could be confusing, like what is a brain scan, what is inflammation, and we will go through all of them during the talk. So just to start with, just to understand why we are doing this type of research. So when we speak about diagnosis of progressive supranuclear palsy or cortical beta syndrome, we really refer to the symptoms and the decline. So when people go to the clinics, they start with some symptoms, and then a clinician confirms the diagnosis eventually. However, behind these symptoms, there is a long cascade of events that happen in the brain, and we call them brain changes because the brain basically doesn't function anymore very well, and we are trying to understand why this happens in people with these conditions. So there are different aspects that happen in the brain. There is a brain shrinkage because the brain cells die over time, and also before then, the cells stop communicating with one another. So we speak about connection breakdown because your cells in the brain needs to communicate all the time for us to be able to function well and perform well. But very importantly, in the last few years, we really started to understand more and more what actually is killing these cells in the brain. And it looks like there are junk proteins or bad bits that accumulate in the brain, and also what we call inflammation. So the brain immune system is not functioning anymore. And there are a lot of research studies also looking at if there are genes involved in these, if there are environmental factors that can influence these changes. So yeah, there is a lot of effort here to try to understand this whole cascade. Something that I focus in particular in Cambridge on for my research is actually looking at these brain changes. And I look at these brain changes because this happens years before the symptoms appear. And what the main aim of trying to develop new things and tools to look at these brain changes is, first of all, identify people that are at risk of these conditions. So looking at early diagnosis and early detections. And also, ideally, we really want to stop these diseases. So slow down the decline, eventually prevent these diseases. And so if we look at what goes wrong in the brain, ideally we can create new drugs and treatments to target those brain changes early on and stop the cascade before symptoms. So how do we look at the brain? Of course, the brain is in the skull, so we cannot look at that with a human eye. And we use different tools. Maybe some of you are familiar with this. The most common brain scan that we use is the magnetic resonance imaging, or MRI. And in my research, I focus also on another scan that looks very similar to MRI, but gives quite different information on the brain. So it's called positron emission tomography, or PET, and it is easy to remember because you can associate this with cats and dogs, but actually we don't look at the animals with this technique. So here is what we see when we use these tools, the PET imaging and MRI scans. This is just pictures of a volunteer in our research. As you can see on the right is an MRI, and with MRI we just take pictures of the brain. It's really like taking photographs, and we can do that over time to look if all bits of the brain are still there, if there is a shrinking process in the brain. As you can see here, there is the eye, and this is the brain. Of course, in the real life, it's not gray, but it's just a way of measuring it with this contrast. On the left, you can see an example of a scan that with PET imaging, the technique that I use. So we basically inject a dye, and these dyes go into the blood and then eventually goes to the brain. And we have very many dyes that are created to target specific proteins, specific parts of the brain, to understand the molecular changes in the brain. So this dye in particular, for example, that we use aims to look at the brain immune system and the inflammation, and looks at this a small protein in the brain, and it goes into the brain and look for the protein, and we can reconstruct these nice pictures, colorful pictures. And in this picture, for example, regions with higher inflammation in the brain are represented with warm colors, so orange or red. And as you can see, there are regions here, like, I don't know if you see my mouse, but there are like spots that are red, and these are like parts of the brain where we can spot inflammation. So what is brain inflammation? I already mentioned that we refer to the brain immune system. So there are a lot of things that we can measure with PET imaging in the brain. First of all, we look, of course, at the neurons, which are the brain cells that help us to function well. So everything that we do, from functioning the world, remembering things, talking, moving, it is all because we have these neurons or cells that communicate with one another, and in the way that they communicate, they allow us to do everything in the world. So in the brain, we also have something that is called macroglia or astrocytes. There are different types of cells, and it doesn't matter to remember the name. It's just to remember, like, you just need to remember that it is a specific part of the immune system that is just specific for the brain, and it's very important for basically keeping the brain healthy and trying to fight with something that is toxic or viruses. And you have to think like for the body, we have a brain immune system that really helps us to fight against anything that can perturb our body. Like, for example, if you cut your hand, you can see this kind of inflammation and the response of the immune system, because the hand becomes painful and swollen and warm. And basically in the brain, there is a similar system. Of course, we don't have a receptor for the pain in the brain, so you're not expected to feel pain in the brain. But basically, something similar happens, and there is like this brain immune system trying to respond to attacks that could be viral or the junk proteins that we see in these in the brain of people with PSP and CBD. However, again, we cannot look at the brain inflammation from outside. So the PET imaging is very useful. This brain scan is very useful to quantify inflammation. And again, like if you see this picture is an example of a person with a condition related to PSP and CBS, and the higher inflammation in the brain, again, is in red colors and orange colors. And if you look at the brain, you see that there are specific regions, like, for example, these are the eyes, and in the frontal lobe, like where there is the front part of the brain, and there is a lot of inflammation. So using these techniques, we looked systematically at volunteers with different conditions, different dementia and different neurodegenerative conditions. We look at a volunteer with Alzheimer's dementia, for example, frontotemporal dementia, but very importantly also volunteers with PSP. as you can see, is something that this response of the immune system is common across all diseases. And like, as you can see, you see orange and red across all these 3 volunteers. But what changes is the specific regions of the brain. There are different parts of the brain that are involved. And based on the parts that are involved by this inflammation, we also see different symptoms across these different conditions. It's very important that inflammation, of course, is not all bad. Like sometimes the brain immune system is trying to be protective and helpful in regards of what happens in the brain and some potential issues that happens in the brains and viruses. The problem is that what we see in people with these conditions, including PSP and CBS, is that the brain immune system is overactivated, is hyperactive, is like always activated, and it becomes kind of a chronic condition having this inflammation in the brain. And this becomes very harmful long term. So it's not like anymore a functional response. It's like something happens in the brain brain immune system gets activated, tries to clean up the mess, and then go to rest. What we see in people with PSP and CBS is that actually the immune system is overactivated, and long-term inflammation cause very damaging changes in the brain. So just to give you an example of how we use these PET scans for research, we recruited the volunteers with PSP in one of our studies, and we look at their brain and inflammation in the brain with this PET scan at the first visit that they came for the research study. And then over time, they came back to our clinics and research facilities for clinical assessment over time. Like every 6 months, in general, they come back. We do a quick test and assessment, and this for many years. So we were trying to understand if what we see at time 0, so the first visit in terms of inflammation, could say something about the future of this in the condition of these people. So as you can see here, the inflammation is present in the brain at visit 0 in volunteers with PSP. And what happens is that over time, so like basically these dots are performance, like how well the person perform or how severe are their symptoms. So higher numbers from 0 to 100, higher number means that the symptoms actually are worse, while lower numbers means that the symptoms are still stable. And as you can see, of course, all volunteers is individual, and there is a great variability between people. There are like volunteers with rapid progression or volunteer with slow progression. As you can see here, there is a steep increase of the severity of symptoms over time for the volunteer on the left, while it's a bit more stable. Actually, after first visit, there is even an improvement because there is lower severity of the symptoms in the volunteer on the right. So we look at how fast these people decline, so how fast people can get worse in terms of symptoms over time. And what we saw is that those people with higher inflammation in the brain at visit 1 were also those people that showed much worse symptoms over time. So this is very important because basically we could picture something in one time, and we could predict who were those people that were declining faster based on the quantity of inflammation in the brain. And this opens up a lot of questions, a lot of potentials as well, because if inflammation is involved in some changes that leads to worse symptoms, maybe actually is a good target to focus on, and maybe something that if we reduce inflammation at the right time, it could help to slow down the getting worse of the symptoms. However, PET scans, I don't know if some of you have been in one of these scans. Sometimes we ask the volunteers to stay for 90 minutes lying down in the scan. And sometimes it cannot be super comfortable. So is something that is not an easy test to do. And also we are quite lucky to have it in Cambridge, but in other countries or in other cities, these PET scanners are not available. So what we are trying to do in collaboration with PSP Association, and they funded this type of research in my lab, is looking at a simple blood test to try to understand if we can capture these levels of inflammation in the blood, and if this inflammation that we can capture in the blood can tell you something about what happens in the brain. Because of course, if we can do that, rather than 90-minute scan in Cambridge, you can do a simple blood test everywhere in the country. So how do we measure inflammation in the blood? So again, I said that there are cells, and it's like in the brains, also cells in the body, and those cells that you see in the blood, they need to communicate. Every single bit of our body and brain all needs to communicate with each other, like us as people functioning in society. And the immune cells communicate with something that is signaling pattern that you can call cytokines. They are chemicals, like I will not enter in the details, but it's just a way of calling each other, saying, oh, you have to go there because there is something that is attacking the body, or you have to enter the brain because there is something that is going wrong, and they alert the rest of the brain. So looking at this signaling between cells and the cells themselves, which type of cells are involved, like is a policeman or like the ambulance, which type of cells are involved, we really want to understand which part of the brain immune system we can switch off, and very importantly also which is the best time window to do that. Because of course, as I said, the immune system can be very useful to try to protect the body and the brain, but we need to identify that time window that actually is not useful anymore, and we can switch it off in the right moment. And of course, identifying the participants and the people with PSP and CBS that can benefit most from these potential treatments. So I spoke about cytokines, and even if I don't enter in specific names of these chemicals, I just want to explain how we try to understand how these cytokines work and which one goes wrong. So you have to think about these chemicals, this communication between cells, as an orchestra. I always use this like metaphor because it really helps to picture how our immune system works. So if you think about an orchestra, if all pieces, all players are good and functioning and well and not sick, they really work well together and they give beautiful music. But of course, if some of them, one of them or some of them don't work in the right direction, of course, you can capture immediately that there is something wrong. And you have to think about that like in a certain way, that the music is, the broken music is something that we can see in terms of symptoms. So we capture that there is something that doesn't work anymore, but we really need to understand which part of the orchestra is that doesn't work anymore, because maybe we can replace it, maybe we can help them to go and feel better and trying to find better music. So these cytokines, we have so many cytokines and we quantify many of them. And because they are an orchestra, we are trying to measure all of them in one go and try to identify what we say that are fingerprints. So here they start to appear some weird names of cytokines, but doesn't matter. It's just these chemicals, they go together and they talk together, and we identified like a pattern, a profile of these cytokines that don't feel together anymore. And we can capture that in the blood. So we basically, if you think about when you go for blood analysis, for other things, you have a list of things that the GP checks on your results to see if you are healthy or not. We are trying to do the same with these cytokines. And what you can see here in the graph on the right, each dot is a participant. So we measured inflammation in the blood of all these participants. And on the average, all people with PSP and CBS had much worse inflammation that we could capture in the blood as compared to healthy volunteers. And also higher inflammation in the blood, we saw that it was related to how bad the symptoms in these people were. So again, inflammation, not just in the brain, but also in the blood, that relates to the inflammation in the brain. related to the clinical severity, so how severe the symptoms are in these people. So again, why we are doing this? I think after these few slides, I hope that I gave you like a good reason to think about inflammation as a potential good target for new treatments. Inflammation happens early on in the disease, and we identified that some tools can really be informative in terms of what happens in the brain, and we can try to scale it up also in what happens in the blood. It's very important to look at both of them because the brain has its own immune system. Of course, the brain immune system interacts with the body immune system, and it is all in constant chat and bidirectional chit-chatting. So just to summarize, we use brain scans, PET imaging. Now, probably when you hear a bit more of these scans, you will know what we refer to as researchers. We are trying to develop new blood tests, and PSP association really has been key in getting the very first few promising results to then expand our research. The combination of these two techniques is really to understand who are those participants or people with PSP and CBS that decline faster over time, so we can forecast the decline based on the quantity of inflammation, but also potentially we can identify who can benefit most from potential treatments. And again, it's very important to be in a good timing to act early on rather than later. So I wanted to thank you for all the attention. Of course, you are very welcome to contact me by email or any other means. There is a group of funding sources that have been quite instrumental, including PSP Association, for the research that we do. As you see, like, it's not just me working on this, it's a big team. On the left, there is my lab, like with very young students and researchers that are very motivated in trying to find a cure for these conditions. And on the right, there is the bigger group of the Cambridge Center for Frontotemporal Dementia-related conditions, led by James Rowe. Some of you probably know him. And of course, I always say that the biggest thanks go to the volunteers, the people with living with PSP and CBS and their families, because you are all the real heroes here to make this possible. So thank you so much. I'm very happy to take any question.
Thank you very much, Maura. That was very clear and nice paced. So thank you very much. So I have a few questions. There's a few questions in the chat, but what I'm going to do is I'm going to take them out and then ask you separately. So maybe the first one from Mike Foster is: is there a difference between PSP and CBD on the PET scans, and I guess by comparison the blood test? Now, you put a graph up for the blood test, but for the PET scans, is there a difference between PSP and CBD for inflammation? And potentially, what he didn't say was whether it would be on the MRI scans as well.
Yeah, so absolutely yes. So what we found in general, both looking at blood tests and the PET scan, is that inflammation is present and very active across both PSP and CBD. However, like what we see in the brain are that the regions involved in these brain changes are different. So like there are different parts of the brain that are involved, from jump proteins, inflammation, and also what you can see on MRI, that is the brain shrinking. And because of that, this leads to different symptoms. So because of different regions involved in the brain, we have different symptoms. But it seems that like actually the inflammation is common across of them. It's just like which regions of the brain are involved that differ.
Perfect. And so that was inflammation. I think someone else, Margaret Stubbs, had said, what's the difference in brain scans for PSP and CBD? So maybe you could talk about that a little bit.
Yes, so like they have very much in common in terms of pathology. So like when we think which are the junk proteins that actually accumulate in the brain and cause the disease, PSP and CBD are quite similar and under the same family, which is a completely different family from, for example, Alzheimer's disease, which causes Alzheimer's dementia. So they are very similar in terms of, again, which are the junction proteins, which are the brain changes in terms of inflammation and brain shrinkage. But again, there is a completely different pattern and topography in the brain of how these pathology spread across the brain. I don't know if, Boyd, you want to add something as a clinician, you know, how
Yes.
you
So
interpret it differently.
so I think what they were getting at was the MRI scan.
Yeah.
Is the MRI scan different? And for PSP, often the stalk of the brain or the midbrain is much smaller and is shrunken a bit more. For CBD, it's often the actual body of the brain, often one side or the other, towards the back called the parietal lobe. So that's often what they are saying. I think just for Siobhan Breyer, she'd said, just a bit of clarification for you, Mara. Did the people with PSP with lower inflammation, i.e. closer to the healthy sample, have fewer symptoms and slower progression?
Yeah, absolutely. What we found systematically is that people with lower inflammation were more similar to what we call healthy volunteers, or like they were, in any case, more stable over time. So they had a much less aggressive worsening of the symptoms.
Okay, and then a hard question from Nigel is, what happened to CCL2? In ALS, it's one of the most consistently altered in blood and CSF.
Yeah, that is a very good question. It's something that we couldn't measure with our assay, unfortunately, because it was part of our panel, but it was not detectable because of the sensitivity. So basically, we started to analyze the blood using something that is very common clinically and available in all hospitals, like the mesoscale discovery. It's like, I don't know if it makes sense for people in the call, but it's like something that has been used for many, many years for clinical assessment, like for other conditions, like autoimmune disorders or cancer. So we wanted to be able to, scale up. So the problem with this technique is that it's not very sensitive. So some of the markers we had to exclude them because they were not capturing many of the participants, also in healthy volunteers. But yeah, that is one of the markers that we would like to look in the future with more sensitive panels, because it could be something that you translate. Something that I didn't say in general for the audience is that there are already drugs targeting the immune system for other conditions. So ideally, if we find that that component of the immune system is involved in PSP and CBD, we can repurpose some of these drugs and try to see if they are effective also in other conditions. So what Nigel suggests is, like look at this marker, because for ALS, there are already, drugs that have been tested.
Good. Maybe related to that, and people haven't asked this yet, but this is my question. For you to bring out, maybe, apart from the drugs to turn off one of those cytokines, could we also try to think about reducing inflammation in other ways? So if people had low-lying chest infections or other things, is that what you're saying about inflammation as well?
Yeah, it's very tricky to disentangle what is peripheral and what is actually very related to the brain. So there are some studies overall, like especially in Alzheimer's dementia, showing that having lower systematic inflammation, so lower inflammation in general, it works like long term for protecting you from dementia. But we really don't know if just switching off the peripheral one for other infections is actually the solution for what happens in the brain. But that would be very interesting. And I think it could be a starting point.
Okay. We've got a few more minutes. So there was one question about the gut. So is the brain affected by the gut, and could chemicals in artificial sweeteners cause brain disease? So maybe you could unpick that question.
Yeah, that's a tricky one. So there is a lot of research actually looking at the gut and brain interaction, especially in Parkinson's disease, because one of the first symptoms is constipation, and it looks like that something that happens in the gut can actually precede what happens in the brain. I'm not aware of any particular studies in PSP and CBD. I don't know if someone else on the call is aware of that. But yeah, there is definitely interaction, but I don't know if there is specific interaction in PSP and CBD.
Okay. And then so there's a few interesting questions. I'm going to try and put them together. So I think we've dealt with, in some ways, we've dealt with Jane Edgington's question about when you fall with PSP, does it cause more inflammation in the brain? But there was an interesting couple of questions from Amanda where she was saying, at the point of rising inflammation in the brain, junk proteins, etc., does that become the focus of treatment rather than the original cause, e.g. bacterial, viral, anything else? So,
Yeah.
and in later stages, you said, you mentioned what treatments aim to contain, reduce inflammation when it isn't severe. What are the research challenges once inflammation is severe? So maybe the question could be about, does the brain start having the disease in one way? So if you think about Alzheimer's disease, the amyloid hypothesis, and then actually the brain's reaction to that precipitates the deterioration in the disease and causes most of the progression. So maybe you could unpick that a bit.
Yeah, that is an excellent question. I think it's the $1 million question. So a bit of what happens first, like it is actually junk proteins like tau accumulating cause a reaction in the immune system, or is actually the immune system that is malfunctioning and cause the junk protein accumulation? The answer is that we don't know just yet. It's something that we are trying to understand. There are a lot of studies in animals, in mice, looking at these, what comes first. But of course, mice are very different from humans in terms, especially of immune systems. So we really need to look at that in people living with these conditions. So one of the things that we are trying to do is like looking at over time with these different markers, like different tools that measure both junk protein and inflammation, and trying to understand what happens first, following them up over time. Is a very tricky question, and there are like also some components, some studies showing that there are some components from the genes, like overexpression of some genes leads to inflammation. So it seems that it's quite early on, but we really don't know what is the cause of this activation of inflammation. It could be that the junk protein starts, then the inflammation gets into the picture, and this everything precipitate. That could be one option, but it could be also that actually inflammation comes first and it doesn't cope anymore and the junk proteins accumulate.
That's great. There's a few questions I'm just going to mop up quickly in the last minute, so you can breathe a sigh of relief, Maura. Thank you very much for all your answers. So there were a couple of questions about what is the best scan for to carry out for PSP, for the best either diagnosis or to look at the interval progression. So usually the diagnosis of PSP is a clinical diagnosis. So it's a clinician looking at someone and spotting the changes that we see and saying, well, look, actually, this is most likely to be PSP or CBD. Scans are helpful to rule out other diseases. They don't really, unfortunately, give the exact diagnosis. And usually we look at patients and people who are in front of us in the clinic, and we look to see how they're progressing, and that helps us to determine progress, which is very individual. Scans like Maura has done are really to try and link the progression with other features of the disease. And just coming, I can't remember who sent this message to me, but they sent it on the chat, so I haven't seen it till late. So please put things on the question and answer session. But with new disease treatments for other diseases, we're having to rehash some of the research that we did at various times in the past, just to try and see if that is helping us to use some of the newer treatments in other diseases in PSP and CBD. Although I understand it is frustrating, research does take some time for people to achieve progress, which is frustrating for people with the disease and the people who are seeing people with the disease, like clinicians and researchers. So hopefully that's most of the questions done. We're going to move on now to Professor Dan Smith. So